Good day, students. I'll be taking you Enzymology, BCH 301. And in this course, it is very important for you to have the copy of notes handed over to your class rep. Please, before you watch or continue with this video, kindly get your notes beside you because I'll be making some reference to some diagrams in your notes which are not present in the video. This is deliberate for you to be able to get your material alongside with the video. Thank you. Now, for in this aspect, I'll be covering mechanism of enzyme catalyzed reactions, effect of temperature, pH, ions, and inhibitors on enzyme catalyzed reactions. What are enzymes? Enzymes are remarkable and specialized protein. Why are they specialized? They are specialized because they are marked to they have unique function or unique role to play. That is, if an enzyme is specialized in converting one substrate to another enzyme cannot do the job. The reason because these enzymes have their unique amino acid that makes them up, while the substrates are of different molecule whereby only one enzyme cannot catalyze all the reactions in the body. That's why enzymes are highly remarkable and highly specialized. They catalyze biochemical reactions that takes place in biological system. They also regulate our metabolic activities. That is, when it is time for your body to require energy, they activate pathway that leads to energy production. And when the energy pool in the body or the ATP pool in the body is sufficient, they also authorize the stoppage, that is conversion of excess food into storage. That's why enzymes are very important in the body. Now, why do you study enzyme? It is essential for you to study enzyme because it helps us to know the cause of any form of ailment, body ailment, so that we can be able to diagnose and also treat any form of disease. It is also used in the chemical industries in where we have fermentation processes. These bacteria or microorganisms or produces enzymes that are being used. These enzymes are produced from microorganisms and they are applied in the chemical industries. We also have in food industries, we also have in agriculture, we also have in preparation of cleaning agent, body care, food preparation. Enzymes are used. And so when we study this enzyme, we will be able to identify where and what role can we apply in them. We also use them in the environmental in the environment, that is in cleaning up or degrading contaminants found in the environment. Now, how do enzyme works? Well, an enzyme works as a catalyst by lowering the activation energy of a reaction, making it more favorable and more easy for a product to be formed. And how does it does it? How does this enzyme carry it out? This enzyme have a pocket called the active site lined up with amino acid capable of holding together the substrate. And when the enzymes and the substrate come together, they form what we know as enzyme substrate complex. And when your enzyme substrate are this when your enzyme substrate complex is formed, then chemical reactions actually carry actually take place. Now, what happens during chemical reactions? We have bonds are being broken, bonds are being formed. It is unstable. It is it is unstable, and as a result of this, product in it. And at the moment your enzyme substrates are formed, it is also called transitional state. That is, it is an unstable state. Your enzyme needs to be recovered and product need to be formed. When you look at the graph, the standard free energy graph, you see a blue dotted line. That is a transition state for uncatalyzed reaction. So actually, for a glucose to turn to pyruvate, it will take a lot of years for it to achieve that. But in the process of bringing an enzyme together, it catalyzes the reaction, it reduces the standard free energy, or that is the free energy, that is energy required for the reaction. It reduces it such that the substrate could be turned to the product. Now, what are theories used to explain enzyme substrate binding or enzyme substrate complex? There are two theories. We have the lock and key theories, and we also have the induced fit uh, model. Now, in the lock and key theory, is just like your door with a key. That is, the lock of your door has been made in with have been made with respect to your key, and so once you dip your key into your lock, it fits in and you can easily turn. Also, this is similar to this lock and key theory of enzyme, that your enzyme and your substrate fits together like key. But the widely accepted theory is the induced fit. It states that the enzyme's pocket are not in a particular shape, or their form are irregular, but upon the approach of a substrate, the enzyme, active site is the active site is formed, and at the end of the day, once the product is formed, the enzyme goes back to its abnormal or to its inactive state, forming another thing, such that no or any other substrate can mimic or will be able to mimic to be able to activate the enzyme in order to convert them to the same product. Now, what are mechanisms of enzyme catalyzed reaction? In your notes, I have number one as acid-base catalysis, covalent catalysis, metal-ion catalysis, electrostatic catalysis, proximity and orientation effect, preferential binding of transition states. And now, for acid-base catalysis, in acid-base catalysis, you have to have the primary understanding of what an acid is. An acid donates proton, while a base abstracts proton, or a base collects proton. So we are told many acid can act as either a base or an acid. For instance, if your enzyme is protein, that means it will donate proton. If your enzyme is an acid, it donates proton to the substrate, and your base, which is your substrate, abstract proton from the enzyme. And so, as a result of this, we have an example which I've given you in your notes, which is not on the slide. It's that of ribonucleases, RNAs. It shows that the ribonucleases works substrate is ribonucleic acid, that is your nRNA. So ribonuclease degrades nRNA. Now I would like you to look at the second diagram under general acid base catalysis in your notes. That's where I will be able to explain. Do not mind the one on my slide. Uh, Now, the acid, the ribonuclease has about 124 amino acid. Now, in the active side of the ribonucleases, there are two important amino acid that carry out this general acid base catalysis. They are histidine 12 and histidine 119. This number stands for the position where this histidine is. If you count from 1 to 12th position, you'll find an histidine there. If you count from 1 to 190 position, you'll find histidine there. Now, this histidine are present in the active site of the end of the enzyme. Another important thing to know is that why histidine is selected or histidine naturally act as acid and base is because its pI value is closer to 7. That is, the pH is almost neutral. So in the mouth of this, as you all know, that ribonuclease acts on ribonucleic acid. And the point of attack, looking at the second diagram, you look at the point of attack is on the phosphodiester bond. Phosphodiester bond. The histidine 12, first, this reaction takes a two-step reaction. First, the histidine acts as a general base, while the histidine 119 acts as a general acid. That is, histidine 12 collects proton from the RNA at the position 2, very close to position 3, holding your phosphate group. And once the histidine withdraws a proton from it, the oxygen becomes readily unstable. And once it becomes readily unstable, it attacks the phosphate group, forming cyclic nucleotide. That is forming 2,3 cyclic nucleotide. As you can see, the arrows showing the reaction. So it forms 2,3 cyclic nucleotide. That's cyclization step. And after this, the important thing is that immediately it attacks a pentose group, or a pentose group has been cut off from the long chain of amino acid. Then, in order for us to get our enzyme back, the histidine 12 have to act as an acid, and histidine 119 has to act as a base, and whereby histidine 12 has to be provide electron, while histidine 119 has to get back electron. And this can be done through the addition of water in your step 2, thereby getting your RNAs back and the substrate being broken. That's all about acid base catalysis. You can look at the keto group in your video. You'll be able to see the arrows showing how the electrons or protons are moving. Look at the acid base, the acid catalysis. Hydrogen is being donated to the ketone, thereby making it uh, making the base more unstable, and it attacks the hydrogen beneath the CH2, and thereby making your enol and getting back to its normal acidic state. The same thing applies to the base. One thing that is very essential in this in this enzymatic reaction is that your enzyme must not go into the reaction. Your enzyme must not be used up. That is, your enzyme disappear. At the end of it, your enzyme has to return to itself while the product is being released. Now, going to the covalent catalysis. Now, it is very, very important that covalent catalysis that happens in the body are temporary covalent catalysis. At times you wonder why will somebody die, or how when they take poison they die. These poisons act form covalent, permanent covalent form permanent covalent bond. What is covalent bond? Covalent bond is the sharing of electron between two elements. And so once this poison gets to an enzyme and form a permanent covalent bond, even when the food or the substrate is available, the enzyme cannot withdraw itself from the poison in order for it to break down food that will give the energy. Thereby, the person dies of stress, cell stress, and a whole lot of cell shock, and the person dies. Also, most times poison works based on permanent covalent catalysis, and thereby killing the patient. But in terms of how a normal body reaction, it is a uh, temporal covalent bond formation. And we have a nucleophile. Nucleophiles are electron-rich group, and while we have uh, electrophile, which are electron deficiency group. I would like to take you back to your notes. That's why I say you have to have your notes in order to be able to have a proper understanding of what I'm passing out. And it is also important that you know that this uh, form of catalysis, that is the covalent catalysis, happens in two stages, have two transition states, as you can see the curve showing that. And look at before I explain what's in your note, you have to look at the diagram here. See the enzyme reacting with the substrate. It breaks one bond, releasing the red substance, holding the yellow one, and the enzyme is released back. Then we have both products. We have the yellow and the red being released. The same thing happens in the covalent in all forms of catalysis. Now let me explain chemotrypsin. What is your chemotrypsin? It is very important for you to know what your chemotrypsin does in order for you to be able to have understanding of the uh, the catalysis. Now there are two forms of there are two stages of this reaction. We have the acylation and deacylation. But there is something you need to understand. uh, Digestive enzymes breaks down polypeptide, that is chemotrypsin breaks down polypeptide. Your chemotrypsin is found in your intestine, in your small intestine, where it breaks down protein molecules, that is breaking down their polypeptide bonds for breaking them into amino acid. And we are told that this chemotrypsin possesses catalytic triad, that is three amino acid working together in order to get this reaction done. And what they do is that the name of the amino acid is serine, histidine, and aspartate. These works together. There is a relay of proton on these chains of amino acid, and they have different position. But you know, in protein structure, your protein have quaternary structures, tertiary quaternary structures, whereby it is folded. And so we have the aspartate closer to histidine 57 and serine 195. And what it does it is that it attacks the carbonyl end of a polypeptide chain. You know, we have the ammonia, the nitrogen end, and we have the carbon end. So this attacks the carbonyl end. It start breaking the amino acid or the polypeptide from there. Now, looking at the diagrams in your notes, especially the one I labeled 1 to 5. 1 to 5, it's very important for you to know this catalysis in that the way they work. Now, serine acting as a nucleo receive serine 195 receive proton from histidine from aspartate sends a proton to histidine. Histidine sent proton to serine, making it a highly or stronger nucleophile, thereby enabling it to attack the carbonyl group to attack the carbon end of the amino acid. And once it attacks it, it forms an acyl group with the amino acid, and whereby the serine group is temporarily cleave from the aspartate and histidine group, and thereby the histidine group releases another proton to the the nitrogen end of the the polypeptide chain, thereby releasing it. Now, after a while, there is the histidine 57 and aspartate 102 activate water molecules, that is, they break down water molecule in order for them to be able to return to get back serine to its natural self. And you can see that the serine, the starting enzyme, is the ending enzyme. So in the process of activating or the addition of water to get back their proton or to get back oxygen to the serine, it now forms hydroxy, it forms bond back with histidine 57, thereby releasing the carbonyl group. So the carbonyl group is released. That's how your protein is being degraded. You can look at the number 1 to 5 steps on how chemotrypsin carry out covalent bonding. Then the last one is metal ion catalysis. Your metal ion catalysis, metal ions, they bind with these proteins in order to stabilize electrons, to stabilize unpaired electrons. To stabilize elements in order for reactions to occur. In your notes, I wrote metalloenzyme. That is, these are enzymes that are tightly bound to metal ions, and we also have metal activated ions. They are enzymes that are activated by the presence of one ion or the other. If these ions are not there, these enzymes will not be able to carry out reaction. So it kind of forms, it says, by in the note I wrote, metal ions participate in catalytic process in three major ways. One, by binding to substrate so as to orientate them properly for reaction. Two, mediating oxidation-reduction reaction through reversible changes in the metal ion state or oxidation state. It's also present in your blood. The ion carries oxygen, and before you know it, it changes from ion 2 to ion 3, and so on and so forth. Then the last one we have by electrostatically stabilizing or shielding the negative charge. That's what your metal ion catalysis does. And so with this, you will be able to have clear understanding how these chemical reactions happen. And it is very, very important that there are various methods. This I mentioned earlier on about 6 uh, form of mechanism by which catalysis enzyme carry out their uh, carry out their reaction. These are based on the peculiarity, the chemical component, that is the amino component of the enzyme. They determine the kind of reactions, the kind of mechanism by which an enzyme will carry out its catalytic activities. Going to the last part of the aspect I will be covering, we have factors that affect enzyme catalyzed reactions. We have your enzyme concentration, substrate concentration, temperature, pH, activators, and inhibitors. For your enzyme concentration, the high, the more you increase your for the enzyme concentration, the more you increase your enzyme, the more you increase the reaction rate. And more enzymes and more your enzyme will collide. That means your enzyme will collide with the substrate. Then we also have what they call the rate of reaction levels up. You can see the curve bending, showing that at a point the enzymes might be the enzymes is more than the substrate, and once the reaction does not increase, it just level up because there are no substrates to bind to the enzymes. Now, substrate concentration, this also goes in the same have the same explanation with enzyme substrate. The more you increase the substrate, the more the reaction rate occurs, and more substrates, more reaction, more collision. And at a time when the substrate is more than the enzyme, the bend, it forms a stationary phase whereby it's no more longer increasing because all enzymes, all the active site present of the enzyme present are engaged are engaged. So that is a maximum rate of reaction. Now, effect of temperature. I don't know. I think I taught you in the one of the other courses that temperature are very important, especially in the extraction of protein. The higher the temperature, the more stable this protein becomes because there's increase in kinetic energy. So, but your enzyme work best at its optimum temperature. If the temperature is 37 degree, when you place your enzyme at the 37 degree, it works best to get its reactions. But any temperature beyond that, it denatures the enzyme. The enzyme lose shape and is unable to catalyze further reaction. We also have effect of pH. The effect of pH change the medium. It says add or remove HF. It disrupts the change. The kind of structural formation, the 3D structure is being disrupted because there are some amino acid that because of the amino acid are uh, acidic. They enjoy the environment they are, and once the pH of the medium changes, they form another shape. Thereby, they are unable to catalyze the normal reaction. Most human enzyme works at pH 6.8. That's almost close to neutral. Any pH lesser or higher than that, those are problem. We have acidosis. That means the person's blood pH is getting more acidic, going to pH of 4, 5. It shows that there is a problem in the body system. We also have presence of cofactors. These cofactors can act as, you can see, these cofactors can act as activators in the body system. They activate, that is, increase reactions and help reactions to get to its end product. We also have effects of inhibitors. Effects of inhibitors. Inhibitors in reactions can inhibit enzymatic reactions, thereby hindering the enzyme from carrying out its natural reaction that is meant to carry it out. And so we have different type of inhibitors. We have competitive inhibitors, we have non-competitive and uncompetitive inhibitors. Competitive inhibitors are inhibitors that have the substrate, the real substrate, and other molecules. They have similar shape, thereby they have they are fighting to occupy the same active site, whereby the non-competitive inhibitors do not attack. It goes to the allosteric's site and have effects on the enzyme. And this is where we stop the course. Please go over the notes and try as much as possible. If you have any questions, look for me or look for your class rep. Also, you can Google online and get more material for better understanding. For better understanding, you can watch more videos on YouTube to have a better knowledge of this course. It is a very, very interesting course, and I believe if you invest more time to it, you'll become a very good student, and you'll be vast in, and you know what you are doing, and you also know how to apply it. I wish you all the best.